Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 5 from our Genetics Essentials, Concepts and Connections 4th edition textbook. This chapter covers linkage, recombination, and eukaryotic gene mapping. So let's go ahead and get started. You need to remember some terms from previous chapters in order to understand what gene linkage is. First of all, remember that principle of segregation of alleles, which Mendel showed us and demonstrated with the monohybrid cross, that alleles separate during meiosis. And then remember that this is the mechanism that's responsible for the segregation of alleles, independent assortment. Alleles at one locus sort independently from alleles at another locus. But what you need to realize is that this only works if the alleles are on different chromosomes, right? Think about it. During meiosis 1, metaphase 1, which is responsible for independent assortment, those tetrads line up down the center of the cell in an independent fashion, right? In a random fashion. That is what's ultimately responsible for the independent assortment and the separation of those alleles. Okay, and that's what allows you to have gametes that have different combinations of these segregated alleles. Remember that. And the other form of recombination during meiosis occurred during crossing over. Remember, during crossing over during prophase 1, you also had some kind of recombination there. Together, recombination and independent assortment, crossing over and independent assortment, accounted for all the genetic variability during meiosis. Now, what this chapter is going to entail is what happens when genes are on the same chromosome. So let's say Mendel did his study, but instead of green or yellow peas and wrinkly and smooth peas being on different chromosomes, what if those two different genes, pea color and pea shape, were both on the same chromosome? Wouldn't that mess up your findings? Wouldn't that result in non-Mendelian inheritance? And the answer is yes. You're not going to get meaningful Mendelian results when two genes are on the same chromosome because it's not going to be independent. In that case, they're called linked genes. Does that make sense? So you have independent assortment when the genes are on different chromosomes. You have non-independent assortment or you have non-recombination usually when they're on the same chromosome. These are called linked chromosomes. Now you do get some recombination because of crossing over, but we're going to get into that. Let's see what happens. So again, here's an example of a normal situation where a parent is homozygous for A and B dominant. The gametes could be AB and cross with a recessive parent, AABB. The gametes can have little a, little b. During fertilization, what do you have? You have heterozygote for both. And then during gamete formation here, if this is a normal situation with the genes on different alleles, what would you expect to see? You would expect to see the original combinations, right? The original combinations, and then you would also expect to see new combinations, like big A with a little b, or little a with a big B, right? So this is what you typically see with independent assortment. This is when alleles are on different chromosomes, right? So through recombination, gametes contain new combinations of alleles. Okay, but one of the first people to demonstrate an exception to this was William Bateson, Edith Sanders, and Reginald Punnett of Punnett Square fame. In 1905, they were following up on Mendel's experiments, and they were simply studying another flowering plant. And what they saw here was that when you cross purple flowers with long pollen, a true breeder, with red flowers and round pollen, let's zoom in here, purple flower, long pollen phenotype, true breeder crossed with red flower round pollen phenotype. You would cross them, and so far so good. All of the offspring were purple with long pollen phenotype. So purple must be dominant. Long pollen must be dominant. So far so good. But in the F2 generation, remember, this is where we expect to see a 9-3-3-1 ratio, because this is a dihybrid cross, two different traits, flower color and pollen shape. We expect to see a 9-3-3-1 ratio of offspring here. And what we see is this. We see a 284, 21, 21, 55. That's really weird. That is not exactly our 9-3-3-1 ratio that we expect to see. We expect to see 9 parts purple long grain, 3 parts purple round, 3 parts red long pollen, and only 1 part out of 16, 1 part red with round pollen. So what's the deal with this ratio? This ratio seems really strange. The highest number is here with the purple and long. The second highest number is the double recessive, which is strange, right? And so obviously when Bateson, Sanders, and Punnett saw this result, they said, well, here's an example of non-Mendelian genetics. Well, if Mendel is correct, why are we seeing this? Why are we seeing this non-9-3-3-1 ratio? Was Mendel wrong? You know, and the answer is, of course, no, Mendel was not wrong. Here's an example of linked genes. Here's an example where 2 different alleles are on the same chromosome. Think about it. If two alleles are on the same exact chromosome, would they segregate independently during independent assortment during metaphase 1? No, they'd be on that same chromosome. So what's the only way to break up that combination in that case? What's the only way to separate this flower color from this pollen shape here? What's the only way to separate it if they're on the same chromosome? Remember, there's only 2 ways to shuffle genes around, right? One's independent assortment. And I just told you here that independent assortment is not shuffling these genes around, because independent assortment, the 2 alleles are on the same chromosome. Does that make sense? And independent assortment just shuffles different chromosomes. What's shuffling the genes here is crossing over. So the only reason you're getting these, you see these new combinations, purple round and red long, the only reason you're getting these combinations is due to crossing over events. And that's why it's so rare. You see 21, 21. So there you go. Crossing over is the only way to get recombinants of linked genes. Okay, very interesting stuff. So again, you're not disproving Mendel. You're just expanding on Mendel's concepts to include linked genes, genes that are on the same chromosome. So if you have complete linkage, what does that mean? That means that you have no recombination whatsoever. Now, here's what you need to understand, and this will become clear as we go along. The closer 2 genes are on the chromosome, let's say there's a gene and then a gene right next to it, the harder it is to have recombination between them. Does that make sense? So the further 2 genes are on the chromosome, the more recombination there is. The closer the 2 genes are on the chromosome, let's say the genes are direct neighbors, the less frequent. Because you have to think like, well, I have to hope that there's a break between this gene and it gets swapped over to the other homologous chromosome, right? Well, what are the odds that that's going to happen if the 2 chromosomes are butt up right next to each other? They're pretty much going to be complete linked. Does that make sense? So you're going to see complete linkage when 2 genes are butt up right next to each other. And you're going to see a higher level of crossing over, not quite the level of independent assortment, but you're going to see a higher level of crossing over with genes that are distant from one another on the chromosome, like physically distant on that chromosome. So it's crossing over with linked genes that leads to recombination here. Here again, you can see where crossing over plays a role. Crossing over takes place during meiosis 1, prophase 1. You see the crossing over event happens. And then during metaphase 1, you've got independent assortment. You've got the tetrads lining up in a random independent fashion. And then you have separation of the homologs. Then you have metaphase 2, where the non-identical sister chromatids line up at the center of the cell, and then you have separation, and you have gametes. And remember, none of the gametes have the exact same DNA. You end up with different gametes. And this is the situation here. Remember, when we're talking about linked alleles, the different alleles are on the same chromosome. So the only way to break up those combinations is to have crossing over between those two alleles. So here's an example. Actually, I thought I had it zoomed in. Here's a test cross revealing the effects of linkage. So in example A, this is an example where genes are completely linked. There's no crossing over happening whatsoever. So let's pretend like genes M and D are right next to each other, right? M for mottled, little m for mottled leaves, and little d for dwarf plant. Okay, and the wild type is dominant for tall and normal leaf plant. So this parent here is a heterozygote. It's a heterozygote. Why? Because it's got big M, big D, little d. Big M and big D are on one chromosome. Little m and little d are on the other chromosome. And you're crossing that plant with a mottled leaf dwarf. In this case, this is a true breeder mutant, which is little m, little d. So if there's no crossing over happening, let's say because, like I said, alleles M and D might be directly next to each other, no crossing over happening, the gametes from this parent are just going to be big M, big D, or little m, little d. You're not going to get another combination. For example, you're not going to get big M, little d, or little m, big D, okay, because it's non-recombinant gametes. This plant, because it's a true breeder, can only make gametes with the genotype little m, little d. So what are the only two types of plants that can form? Well, the only two types of plants that can form are heterozygotes, like this parent here, big M, big D, because it got this gamete fertilizing the little m, little d gamete, and that forms a heterozygote. The only other option is the little m, little d gamete fertilizing the little m, little d gamete, and you're getting a mutant. So you should get half heterozygote and half homozygous recessive offspring. Okay, again, this is an example of completely linked genes. And when does that happen? It tends to happen when genes are really close together on the chromosome. Different alleles are really close together on the chromosome. Now, if genes are unlinked and they assort totally independently, okay, this would be, like I said, if the genes are on different chromosomes, for instance, then you would expect a completely different outcome from this cross. Because look, here you have the heterozygote parent and the homozygous recessive parent. The homozygous recessive can only make gametes with little m, little d. However, in this case, the heterozygote parent can make all kinds of different gametes. Big M, big D, little m, little d, big M, little d, little m, big D. Why? Because you can mix and match these different alleles because of independent assortment, because the alleles are on different chromosomes. M and D are on different chromosomes. So you're going to get a quarter, this variant, a quarter, a quarter, a quarter. In this case, after fertilization, you're going to end up with all kinds of progeny. Non-recombinant progeny, which means progeny that look like the parents, and you're going to get an equal proportion of recombinant progeny. These are combinations that you did not see in the parents. Does that make sense? So you can see there, Mendel got really lucky because all of his studies with the pea plants, the pea shape, pea color, pod shape, pod color, all the stuff he studied had to do with independent assortment. He got lucky, and the traits he was studying had complete dominance, so a clear dominant and a clear recessive trait, but they also were on the different chromosomes. If they were on the same chromosome, that would have given his studies, they would have introduced a lot of confusion in his studies, and he wouldn't have gotten as clear and concise results as he did. They would have been kind of confounding factors that would have made things a little bit confusing or surprising. Right? Does that make sense? So again, you can see here, in the case of linked alleles, linked genes, the only way to shuffle them around is by crossing over. If the two genes A and B are too close together, then there's going to be no crossing over usually between the two, and you're not going to have any recombination. Each gamete receives a non-recombinant chromosome. But if the alleles are separated by enough distance, the chances go up for recombination. But remember this, recombination during crossing over, you know how in crossing over you have a tetrad, so 2 and 2, right? A tetrad, only one sister from this homolog exchanges information from one sister from the other homolog. Does that make sense? So you've got a tetrad, only 2 of those homologs are exchanging information. The other 2 homologs stay non-recombinant. They do not engage in crossing over. That's good to know. That's something you should know. So look here. Here you have a tetrad. These 2 middle homologs, the red and the blue middle homologs, are engaging in exchange. They're engaging in crossing over. So they're going to have recombination. Now, this red one's big A, little b. This blue one's little a, big B. However, notice that the outer red one and the outer blue homolog does not engage in crossing over. This is always the case. Crossing over only occurs with one homologous pair in the tetrad. So just be aware of that. Okay. So that's why it says here, a single crossover produces half non-recombinant gametes and half recombinant gametes. And like I said, the further the alleles are on those chromosomes, the more likely they'll get recombined. The closer those alleles are on the chromosome, the less likely, to the point where it's rare to see a recombination, and they call those non-recombinant alleles. All right, so concept check time. For single crossovers, the frequency of recombinant gametes is half the frequency of crossing over because frequency of crossing over because, let's go through these one by one. A test cross between a homozygote and heterozygote produces 1/2 heterozygous and 1/2 homozygous progeny. No, that's not true. The frequency of recombination is always 50%. That's not true. Each crossover takes place between only 2 of the 4 chromatids of the homologous pair. Remember, that's what I just said. You have 4 chromatids in the tetrad. Only 2 of them are doing the crossing over. So that's true. And crossovers occur in about 50% of meiosis. That's not true. It has to be C, right? Again, I told you, when you have a tetrad formed, only 2 members of that tetrad, 2 homologs, are engaging in crossing over. This is why, for a single crossovers, the frequency of recombinant gametes is half the frequency of crossing over. So now, what is the frequency of crossing over? How do we calculate the frequency of crossing over? Now we can engage in some math, right? And keep in mind, these studies were pre-genomics, solving the genomes for the human. We've solved the genome for the fly. We've solved the genome for the worm, the C. elegans worm. We've solved the genome for all of the model organisms and more. But back when these studies were done, we hadn't solved the genomes. We didn't know how to sequence the chromosomes and sequence all of the DNA inside of your genome. So instead of using sequencing, what they would do is look at recombination frequency to determine where genes would lie in relation to one another and on the chromosome. Think about it. I told you earlier, the further alleles are on a chromosome, the more likely they'll recombine. So by doing tests, by doing crosses over and over, you could start to determine, well, this gene and this gene are really close together, but then this genes must be really far from this gene, but because it recombines more frequently, and it has to be on the same chromosome because it doesn't assort independently. So you could actually start mapping the genome. You could actually start mapping the chromosomes by looking at these genetic crosses that were done with linked genes, and then you could actually start predicting where the genes lie along the chromosome based on these recombination frequencies. So how do you calculate recombination frequency by doing these crosses? It's the number of recombinant progeny over the total number of progeny times 100. Now, there's also, you have to factor in coupling and repulsion, which I'll explain in a little bit, but coupling is simply called the cis configuration, where the wild type alleles, the plus alleles, are found on one chromosome and the mutant alleles found on the other chromosome. So remember this. This would be the example here, where the wild type is found on one chromosome and the mutant is found on the other. That's what we're talking about here, about coupled configuration. Repulsion would be the opposite. Repulsion would be when you're starting with big A and a little b, or the wild type A and the mutant B on the same chromosome. Coupling is when you start with the wild type on the same chromosome. It's just a terminology thing that will help you to understand these studies. So here we're looking at crossing over between linked genes producing non-recombinant and recombinant offspring. So this would be linked gene study of the modeled plant. Remember mottled and dwarf? We were looking at this earlier. So here's what we would see. We start with a heterozygote dominant, which is big M, big D, little d. Now, is this the coupled or the repulsion? Is this coupled or repulsion? This is coupled configuration, right? Because the big M and the D are on the same chromosome. So this is a coupled, and here this is coupled as well, little m, little d. So this heterozygote is crossing with this homozygous recessive. Remember, there's gamete formation. There are non-crossover gametes, big M, big D, little m, little d, and then there's crossing over that can happen too, right? Remember, crossing over can introduce recombination, and that recombination would give you half non-crossovers, remember? Because out of the tetrads, only 2 are doing crossing over. So you get half non-crossovers and half crossovers, big M, little d, little m, big D. So these are your pool of gametes here. And so what are the possible configurations? Well, I would say, okay, what happens if this gamete fertilizes this egg, this gamete fertilizes that egg? And so you can break it down into, you could predict the outcomes. So this is what we see in the progeny here from self-fertilization. We see 55 non-recombinant heterozygous dominant. We see 53 non-recombinant, just like the parents. So non-recombinant means same phenotype as the parents. So remember, heterozygous dominant and homozygous recessive. We saw 55 like the first parent, 53 like the second parent. But then we did see new combinations. We did see recombinations. But look, we see only 8 recombinations that are big M, little d, little m, little d. So these are normal leaves but dwarf plant. And we only see 7 that are mottled leaves tall. Okay, and so the reason the fact that these ratios aren't 1/4, 1/4, 1/4, 1/4, the fact that you're not seeing an equal amount of all of these should automatically suggest to you that this is not independent assortment, that there's linked genes going on here. And the linked genes, and then what you could do is calculate the combination frequency by doing math with these numbers. So again, the conclusion is with linked genes and some crossing over, non-recombinant progeny predominant. So when you have linked genes, you're going to see much more frequently, you're going to see non-recombinations, combinations that were present in the parents. Okay, so again, what's coupling or the cis configuration? This is where one chromosome contains both wild type alleles and the other chromosome contains both of the mutant alleles. And again, I told you this is an example of the coupling configuration or the cis configuration. Repulsion would be the trans configuration where the wild type allele and a mutant allele are found on the same chromosome. So now let's take a quick break time with Gizmo and Wicket so I catch my breath, and we'll come right back with more. Hey everyone, welcome back from break time with Gizmo and Wicket. So where we left off, I was telling you a little bit about the coupling or cis configuration of alleles versus the repulsion or trans configuration of alleles. And this does factor in when you're doing these types of genetic crosses. Let me give you an example. If you start with the cis configuration with two alleles, let's say P plus and B plus, this would be the wild type green thorax and brown puparium, and you cross that with the mutant purple thorax black puparium. If it's cis configuration, well then after the cross you're going to see a majority of the progeny are going to exhibit the cis configuration. They're going to be P plus B plus or green thorax brown puparium, and they're going to also mimic the other parent. They're going to be purple thorax black puparium, and that makes sense. However, you could do the exact same looking cross. You know, you're starting with green thorax brown puparium just like before, purple thorax black puparium just like before, but if these are cis in this case trans in configuration, where you have got P plus B, and on the other chromosome P B plus, this would be the trans orientation. Because of this, in the progeny you're going to see what appears to be recombinants with green thorax black puparium and purple thorax brown puparium as the most numerous progeny. It's going to be the what appears to be a crossover but really isn't is going to dominate in the progeny number here. So the conclusion you would draw is that the phenotypes of the offspring are the same, but their numbers differ depending on whether alleles are in coupling or in repulsion. Just keep that in mind, because you could do what appears to be the same cross, but because of cis, the non-recombinant progeny look one way, and because of trans, the non-recombinant progeny could look a completely different way, almost as though there was recombination. So let's do another concept check. The following test cross produces the progeny shown. A, the big A, big B, little b, crossed with little a, little b, little b. And here's the numbers we see. All right, the first thing that should draw your eye is these big numbers here. 40, little a, big B, little b's, 40, little a, big B, little b's, and only 10 of these other types. So the question is, were the A and B alleles in the heterozygote, the big A, little b, big B, little b parent, in coupling configuration or in repulsion configuration? Well, let's take a look at the most numerous progeny. Remember that this parent, the homozygous recessive parent, has to donate an A and a little b, a little a, a little b. So it looks like from this parent you got little a and big B, the most common. You see little a and big B. So what that should tell you, little a, big B, is that this parent is in repulsion or trans configuration. And that is the answer, repulsion. Now moving on, how do we tell where these genes are on the chromosomes then? If we wanted to figure out, okay, we know there are two alleles on a chromosome. Where about on the chromosome are they? How far are these alleles on the chromosome from one another? We can actually construct a genetic map using what's known as a 2-point test cross or a 3-point test cross, and I'm going to explain those in just a second. But we can actually look at distances between these alleles and the order of these alleles by doing test crosses, by doing these types of test crosses. And the units, by the way, that they use for distance between alleles is either in what are known as map units or centiMorgans after Thomas Hunt Morgan. One thing I need to explain before I explain these crosses, though, is that we've talked about crossing over during meiosis 1. Remember in prophase 1 you could have crossing over in a tetrad. Keep in mind, crossing over only happens between 2 of the homologs out of 4. Remember that 4 total chromatids come together in a tetrad during crossing over in prophase 1. Only 2 of those chromatids exchange information. So one blue chromatid will exchange information with one red one. That's always the case. This red one and this blue one on the ends are not going to exchange information, and that's always the case. So rule 1 of crossing over is only 2 of the homologs out of the tetrad cross anything over. Rule 2, you should know, is that you could have single crossover events. You can have double crossover events as well. So keep in mind, you can have a double crossover. And sometimes this double crossover can happen, and you might think there was no crossover happening at all, because take a look at this. Let's say there's a crossover here. You got alleles A and B separated by quite some distance. Let's say there's a crossover event here and then a double crossover here. Well, there's no actual recombination between the A and B alleles, is there? However, you did exchange some actual genetic material. So in this case, you might think this is non-recombinant progeny. However, parts of the chromosome have recombined. Just be aware of that. So concept check number 3 time. How does a genetic map differ from a physical map? Like I just told you, a genetic map is done by doing these 2-point or 3-point crosses with actual breeding of these different phenotype parents. A physical map is actually looking at the genetic sequence, which is what we can do nowadays, by the way. Ever since the early 2000s, we've known how to sequence genomes. We sequence the human genome, we sequence the fly's genome, we sequence worms, we sequence all the genetic model organisms have been sequenced. So we can go in and tell you exactly how far this gene is from that gene in a number of bases in between. But back then, in the times of Thomas Hunt Morgan, for instance, we didn't know how to sequence the genome. So people would figure out the distance between genes by how often the recombination was occurring. And if the recombination was occurring more frequently, you could tell that the alleles are more distant on the chromosome. And if they're more closer together, the recombination happens less frequently. Let me show you something. Look at this. Imagine if allele A and B were right next to each other, like butt up, right next to each other. What do you think the odds are if A and B are super close together on the chromosome? What are the odds that the crossing over event is going to happen in between A and B? probably not that common. Does that make sense? But what if A and B on the chromosome are really far apart? A is way up here in the chromosome, B is way down here near the tail, near the telomere. Well, then crossing over is going to happen a lot more frequently. Does that make sense? So by using 2 and 3 point test crosses, you were able to figure out where these different alleles are along a chromosome. But now with the physical maps, gosh, we've got the genetic sequence done. You could go and look it up with a computer, a few clicks of a mouse button, you'll know exactly where these different genes are. So again, genetic maps are based on rates of recombination by doing these test crosses. Physical maps are based on physical distances that you can actually sequence now that we know how to sequence genetics, genomes, I should say. So again, why do you do these test crosses? You do them to determine the gene order and determine the location of crossovers. So let's look at what these crossovers can show us. We can have a crossover event here. So you've got alleles A, B, and C on this homolog, let's say the maternal homolog pair, and then you've got alleles little a, little b, and little c on the paternal homologous pair. Remember, only 2 of these exchange information. Only 2 of these do crossing over during prophase 1, so only these 2 will. Now, you could have crossover here, right? You could have a crossover event here, and that would be this middle example where the big C and the little c swap over, right? That's a single crossover event. You could have a crossover here. You see this here where I'm kind of indicating? You could have a crossover here, and that would result in the big B and the big C swapping with the little b and the little c. You know, you could have that. Or do you remember what I said earlier? You could have a double crossover where there's a crossover event here and a crossover event here. And in this case, the B, the big B and the little b will swap, right? So you'll have big A, little b, big C, and only the B's have crossed over. So that would be a double crossover. Now again, do you remember what I said earlier? Which one of these 3 examples would be the most rare? That's right, the double crossover would be the most rare. You're going to see that the least number of times if you do these genetic crosses with, if you're monitoring 3 different alleles, you're going to see this event happening the least frequently for the middle allele, because it requires a double crossover event to occur. And double crossovers don't happen every time, right? So conclusion, recombinant chromosomes resulting from the double crossover have only the middle gene altered. So let's take a look at one of these 3-point test crosses. And remember, why do we do these? Well, to figure out the sequence of genes, to figure out what's the middle gene, right? By the way they have these genes listed with S, T, E, and SS, that's not necessarily the order here. This order is arbitrary. S, T, E, S, S, this is arbitrary. We're trying to figure out what's the middle gene here, okay? So just keep that in mind. So here you're starting with 2 flies, 2 parent flies, a wild type fly with ST plus, E plus, and SS plus. And it looks wild type, but it's actually a non-true breeder, isn't it? It's a heterozygous parent. It's a wild type on 1 chromosome, so it's cis configuration on 1 chromosome, and it's mutant on the other chromosome, but it appears wild type because the wild type alleles are dominant. And then the other parent is homozygous recessive. Now we do our test cross, and that's how you set up one of these 3-point test crosses. Keep in mind that we have to break down what these alleles are. So ST plus means red eyes, that's wild type. E plus is gray body color, that's E plus wild type. And SS plus is normal bristle, that's wild type. Okay, so we do our cross, and then we count our progeny. We count our progeny and look at the phenotypes. Look at this. These are the 2 most common progeny, right? You see these huge numbers of progeny here. These are the 2 most common. And no surprise, these are the non-recombinants. You see, all wild type. So there was no recombination between this chromosome and this chromosome. You see ST plus, E plus, SS plus, there was no recombination. And all mutant, you see there was no recombination here either. There was no recombination here either. So these with the largest progeny number, those are the non-recombinants. Okay. Now look at the other combinations. There's 1, 2, 3, 4, 5, 6 other combinations. And 4 of them have bigger numbers. Look at this, 50, 52, 43, 41. Now look at this. These two, spineless, where only SS swapped over. You see you have ST plus, E plus, but SS swapped over. And scarlet ebony, again, where only the SS swapped over. You had ST, E, and then SS plus. So it's something about this SS or SS plus allele, something about that. Look, it swaps over, it recombines very infrequently. Well, perhaps, what would you say? Perhaps that's our culprit. Perhaps that's our middle allele. And you'd be right. You'd be right. So to figure out what's the middle allele, you figure out what is the least common progeny and what recombination event does that align with. In this case, it's SS recombination, where this guy, this SS plus, swapped over with this SS. That's the only explanation for these two flies. And that is your double crossover. So I'm going to show you that in just a minute after these concept checks. So concept check time. Write the genotypes of all the recombinant and non-recombinant progeny expected from the following 3-point cross, which is very similar to the example we were just covering. This is your heterozygote parent. Here's your true breeder homozygous recessive parent. There's not really any recombination that can happen in this parent, so we're looking at only this parent. So you could have no recombination. No recombination would be M plus, P plus, S plus. M plus, P plus, S plus. The other non-recombinant event would be MPS, MPS. And then you can have any other combination. You could swap the S's around. Let's see, where's the one where we just swapped the S's around? This one? You can, there should actually be a plus here. It's kind of missing. You could just swap the P's around. You could just swap the M's around. So there's 8 different combinations, right? There's 8 different combinations. Just like here. Let's go back. Just like here, there's 8 different combinations. You're going to get your 8 different combinations here. Okay. Here's another concept check for us. A 3-point test cross is carried out between 3 linked genes. The resulting non-recombinant progeny are S plus, R plus, C plus, and SRC. These are the non-recombinants. So those should be the most common. And the double crossover progeny are SRC plus, S plus, R plus, C. So which one is the middle locus? Which one of these alleles is in the middle? Well, it looks like, the only one that doesn't belong is this C. It's the C plus has, so this C has swapped with that C plus. You see the C's have swapped places, so C locus is the middle locus. And that's going to give you the least number of progeny in these crosses, these 3-point test crosses. So going back to that example from before, we were correct. Remember we said SS was the fishy one. SS was the gene that swapped over that gave us the least frequent progeny number. Well, that is the middle allele. And the other alleles are at the ends. And just to wrap up this chapter, we can take a look now at the whole genome. Remember, Drosophila melanogaster, the humble fruit fly, is a genetic model organism. It only has 4 chromosomes. Chromosome 1 is the sex chromosome, or an X chromosome. And then you have chromosome 2, 3, and 4. Do you remember on Thomas Hunt Morgan's early fly experiments with the white eye phenotype. Remember how it was on the X chromosome of the fly? That's interesting to think about. So this is what we know now, and obviously, again, nowadays a lot of these old studies are antiquated and no longer really necessary for mapping out genomes, because nowadays we can just sequence. We have the sequence of so many different species of organisms now, not just the human and the other major model organisms, but we're sequencing pretty much everything nowadays: plants, flies, insects, animals, extinct animals, etc. And again, just to review, if two alleles are really close together, you see like sepia eyes and hairy body, if these two alleles are super close together, do you think there's going to be a lot of recombination that occurs between these two? Or are they going to appear more linked? Are they going to show non-recombination? I would say if you're going to see a sepia eye fly with hairy body, it's usually going to be always the case. You're not going to see too many recombinants between sepia and hairy. But what if we're looking at hairy versus veinlet veins? Well, look how far hairy is from veinlet. There's a lot more likelihood that there's going to be a crossover event here during prophase 1. So you're going to get a lot more recombination between veinlet and hairy than you would between sepia and hairy, because what's the odds that a crossover event's going to happen between these two little this tiny distance between these two alleles? So think about it that way. Interesting stuff, isn't it, to think about. A relatively short chapter, but a good one, a really interesting one. Let me know if you have any questions, and I'll catch you guys next time.
Dr. D, 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 Dr.